The respiratory system is an organ system in the body that functions in gas exchange with the environment. Exchange of gases like carbon dioxide and oxygen are essential for sustaining life forms. Oxygen is necessary as an electron acceptor in aerobic metabolism for oxidative phosphorylation, or the synthesis of ATP, in the electron transport chain. ATP is the energy source needed for muscular contraction in mammals. Therefore, oxygen must be readily available for ATP synthesis for use by metabolically active muscles. Since muscles need large quantities of oxygen, it is transported by proteins in the blood and delivered to muscle tissue for storage. One of the proteins that does this job is myoglobin. Myoglobin can exist in two forms, deoxymyoglobin shown to the left and oxymyoglobin shown to the right. Myoglobin is a hemoprotein found in the skeletal muscle of mammals that functions in oxygen storage and diffusion. A hemoprotein is a protein that contains a heme prosthetic group with an iron 2 ion at its core. Myoglobin is made up of one globin subunit, which are units consisting of 8 alpha helical structures. A single polypeptide chain forms this alpha helix by the hydrogen bonding interactions of the 153 amino acids in myoglobin. Because deoxymyoglobin and oxymyoglobin contain 5 and 6 occupied coordination sites around the iron ion, respectively, the metal centers maintain two different geometries. When the iron ion is deoxygenated, the heme group maintains a square pyramidal geometry shown here. This means only 5 of the 6 coordination sites are occupied. When the iron ion is oxygenated and all 6 coordination sites are occupied symmetrically, the heme group maintains an octahedral geometry. As previously stated, the function of myoglobin is to store oxygen. This occurs at the heme prosthetic group, which is bound to myoglobin by a proximal histidine residue. The heme group is the metal center I will be focusing on for the remainder of this video. Heme is a polydentate structure made up of a large aromatic porphyrin ring with 4 pyrrole nitrogens and a ferrous or iron 2 ion at the center. The affinity for iron 2 as the metal center in this complex can be explained by the chelate effect and hard-soft acid-base theory. The chelate effect refers to a multidentate ligand having an increased affinity for a metal ion in comparison to monodentate ligands. An increased number of ligands results in an increase in entropy, making the overall structure more stable. The porphyrin ring of heme is a tetradentate structure. Heme, along with the nitrogen from the imidazole of the proximal histidine residue, serve as chelators for the iron 2 metal ion center. Having these chelators in the metal complex leads to an increase in entropy and therefore an increase in stability. Thus, the iron ion is held very tightly within the heme group. The hard-soft acid-base theory classifies metals and ligands into different categories based on their character, including charge density and metal-ligand complex interactions like covalent bonding and electrostatic interactions. The general trend for interactions between metals and ligands is like prefers like. This means that hard acids prefer to bind with hard bases, soft acids with soft bases, and borderline acids with borderline bases. Iron 2 is a borderline acid. It is held in place by nitrogens from the porphyrin ring and the imidazole of the histidine residue. Nitrogen is typically categorized as a hard base. However, due to the high polarizability of the porphyrin ring and imidazole histidine in heme, the nitrogens are considered borderline. Thus, the borderline nitrogens prefer to interact strongly with the borderline iron 2, again allowing for the iron to be tightly held within the heme structure. Moreover, in terms of kinetics, the iron 2 ion held within heme is very inert. According to Coulomb's law, an increase in oxidation charge means a decrease in lability. Therefore, iron 2 is less likely to release the nitrogen ligands. This also explains the high affinity for iron 2 in heme. Now let's move on to the function of myoglobin, oxygen storage. Oxygen binding occurs at the iron center of the heme group. Let's consider the 18-electron rule regarding the likeliness of an additional ligand to bind. In deoxymyoglobin, the total electron count is 16 electrons. There are 10 electrons from the 4 nitrogens of the porphyrin ring and histidine residue, and 6 from the metal D electrons, adding to 16 electrons. Therefore, the metal ligand complex is available for another ligand to bind. Since the function of myoglobin is the storage of oxygen, electron counting determines that the heme complex is available for another ligand, this case in the form of oxygen. The oxidation state of iron in oxymyoglobin is a highly debated topic because data is inconclusive and seemingly contradictory. Based on data from IR spectroscopy and magnetic measurements, scientists have proposed 2 possible modes of oxygen binding to the iron ion. First, the dioxygen molecule binds to an iron 2 ion but does not transfer its electrons and remains an iron oxide as shown here. Second, the dioxygen molecule binds to an iron 3 ion and shares its electrons, forming a superoxide. There is data that supports oxygen binding and forming a superoxide. A study looked at dioxygen species and their vibrational properties and bond length via IR spectroscopy. They found that oxygenated myoglobin has an oxygen-oxygen bond vibration frequency that is closely related to the oxygen-oxygen bond vibration frequency of superoxide. This IR spectroscopic data is taken as evidence that myoglobin might maintain an iron 3 oxidation state when oxygenated. However, based on the magnetic measurement of iron 2, it is assumed that oxygen binding to iron 2 is preferred over iron 3 binding. The ligand field theory suggests that deoxygenated myoglobin is paramagnetic because there are 4 unpaired electrons. The energy diagram of the square pyramidal iron 2 is shown here. Notice the 4 unpaired electrons in a high spin configuration. When oxygen is available to bind, there are 2 unpaired electrons that can interact with the iron 2 in either a strong sigma bond or a weak pi bond. The resulting iron-oxygen complex in an oxygenated myoglobin becomes diamagnetic as all electrons are now paired. This suggests that oxygen binding to the iron 2 ion is favored over binding to the iron 3 ion, as demonstrated by the diamagnetic property of the iron 2 oxygen complex. The binding of oxygen to the iron 2 ion can also be explained by ligand field stabilization energy. A high-spin octahedral iron 2 has an LFSE of negative 0.4, meaning it is not very stable, and the increase in lability allows for a ligand to bind easily. Oxygen can bind to the iron 2 ion, which would allow electronic rearrangement to a low-spin iron 2 complex, which is much more stable. Despite the high-spin nature of the iron 2 ion in heme, the interactions between iron and the porphyrin ring and oxygen create an overall low spin for the system. Thus, the iron-oxygen complex would have 0 unpaired electrons. One human disease that is associated with myoglobin is rhabdomyolysis. This is characterized by the breakdown of muscle tissues, resulting in the release of muscle fiber contents like myoglobin into the blood. When this occurs, myoglobin is filtered out of the body by the kidneys, but in doing so, myoglobin is broken down into substances that can damage kidney cells. In summary, myoglobin is a protein in the blood that functions in oxygen delivery and storage into skeletal muscles in mammals. The stability of the heme structure of myoglobin is explained by the chelate effect, HSAB, and kinetics, while the binding of oxygen to an iron 2 ion is explained by the 18-electron rule, ligand field theory, and LFSE. I hope you all learned something today, and thank you so much for watching this video.